nabili nyo from MedTest. So I'm going to discuss about the principles of real-time polymerase chain reaction technique to be used for COVID-19 detection or the detection of the virus SARS-CoV-2. So for this discussion, if you have questions, you can just cut me off. It's fine. Or you can type in your questions on the chat box. Then titignan ko siya. So I hope lahat po ng participants ay nandito na. And also I have with me my colleagues. I have Mavil, Soriano, and Clean Rose. Okay. So I'll be starting na. Okay. So, yun. So basically ang discussion po natin ngayon is more on the principles. So the concepts of are the concepts that underlies all the instruments that you are going to use to detect SARS-CoV-2. So, ang scope po ng discussion natin ngayon ay magsa-start po tayo sa introductory about the pathogen, the SARS-CoV-2, and we'll go with an overview of the diagnostic workflow for SARS-CoV-2 detection. And then I'm going to talk about the principles on the sample preparation. So sa sample preparation, included po dito ang extraction. So extraction using an extraction kit and the automated or the instrument, the Kingfisher Flex system. And then next we'll tackle also the polymerase chain reaction technique, so or PCR in short. So ito po yung technique na ginagamit natin to detect the SARS-CoV-2. And then after that, we'll go with the principles of real-time polymerase chain reaction or qPCR or RPCR. So real-time PCR, kung anong difference niya dun sa the conventional polymerase chain reaction technique. And then lastly, how to read the results of a real-time PCR instrument. So let's start. So basically, SARS-CoV-2 is short for severe acute respiratory syndrome coronavirus 2. So we have the SARS-CoV way back a decade ago yata. So this is a new strain, no? Which is a small virus ranging from 60 to 140 nanometers in diameter. So ganun siya kaliit. And it belongs to a family of coronaviruses that infect both humans and animals. So ang typical na characteristic ng isang coronavirus is that it has its own genetic material in the form of an RNA or ribonucleic acid. And this genetic material is enclosed in a capsule made of protein and fat. So the target of this virus, when it gets to its host, it infects mainly all of the cells in the epithelial system. So like we have the tracts, the blood vessels, it also infects heart, lungs, and kidneys. So, but mostly um, in order for us to detect the virus infecting a human, we usually collect um, samples from the nasal area and the mouth area. So, um, so we know that the genetic material of the virus is in the form of RNA. So, RNA um, is a nucleic acid. So, what is a nucleic acid? Nucleic acid is a biopolymer present in uh, almost all of the living organisms. So, hindi lang uh, tayo ang merong genetic material in the form of nucleic acids, meron din ang virus. And nucleic acids can either be a DNA or an RNA. So kung makikita nyo, uh, I hope nakikita nyo yung cursor. So this is the structure of your DNA and compare it with the structure of your RNA. So pretty much they're both different. You can uh, illustrate DNA as like a ladder, but the RNA is like half a ladder. So basically, the DNA is composed of two strands of nucleic acid, while the ribonucleic acid or RNA is composed only of one. Now, tinatawag po siyang uh, biopolymer because this molecule is made up of nucleotides, which is the building block of the nucleic acids. So ito po yung, I'm not going to discuss it in detail, but this is the, sorry, this is the structure of your nucleotide. And this is important because the nucleic acids are identified based on the nucleotides that it contains. Usually, um, scientists or researchers would want to identify the order of the nucleotides that is present in the nucleic acids. And this orderly form of nucleotides is what we call a sequence. Now, the basic difference from an RNA to a DNA is that an RNA contains uracil, while the DNA contains thymine. So ito po yung mga differences ng structure ng DNA and RNA. Aside from that, the RNA is a single-stranded molecule. So, now this is just a graphical representation of your SARS-CoV-2 virus. So kung makikita nyo, the it's depicted as a globular particle, no? And it's made up of proteins. No? We have what we call spikes. So ito po yung importante kasi ito yung responsible for the virus to infect the cells. And then enclosing uh, the genetic material of the SARS-CoV-2, no? And then inside the structure, makikita natin yung genetic material ng virus, which is an RNA. So, yung RNA po, yun po yung uh, nag nagko-contain ng information about the proteins na kailangan to express the whole virus itself. So, so for the COVID-19 detection workflow or SARS-CoV-2 detection workflow, we follow a series of steps, from sample collection to data analysis. So in sample collection, we basically collect materials from a patient or human, for example. And it can be from blood or from fluids, from tissues, or usually um, we have to know if we're dealing with a certain virus, we have to know where the virus could possibly be thriving in the human body. So doon natin siya kukuhanin. And then um, after collecting the sample, we proceed with sample preparation. So in sample preparation, yung yung nakuha po nating material from a patient is processed chemically and physically so that we would obtain a target material. So in the case of COVID-19 detection, what we are after is the genetic material of the virus, which is the RNA. Then after we get or we obtain the RNA, we proceed with the detection. So in the detection step, we use an equipment that is able to tell you whether or not you get a, you get your target genetic material from your samples. So in this case, for COVID-19 detection, the instrument that we use is a real-time polymerase chain reaction instrument. And then once we are done with the detection step, we would proceed with the analysis and interpretation. So basically, we want to verify whether the results from the detection step is valid or not. So for sample preparation, this involves mainly uh, these steps. No? So the first one I want to discuss is the deactivation of viral particles. So may mga laboratories na nagkakandak or has their own protocol in deactivating the virus prior to processing them. This is to uh, reduce or prevent the analyst from getting infected by the virus. So this depends on your laboratory um, kung meron po kayong ganitong protocol or not. Then the next is cell lysis. So part of the sample preparation is the cell lysis. So after we've collected our samples from a patient, we target to break up or disrupt the cells that we collected so that we will be able to get the target material. So in the case of COVID workflow, we want to get the RNA that is inside the cells. Now, cell lysis is a very important step in the sample preparation because it will determine if your protocol is effective kasi pag hindi na-lyse o pag hindi na-disrupt yung lahat ng na-collect mong cells from your patient, there is a possibility na konti lang din yung makukuha mong target material or RNA. So in this step, or in this concept, the target quantity is being addressed here. Then the next, which is the extraction step. So the extraction step is a combination of a chemical and physical techniques in order to separate the target material, which is the RNA, from other cellular materials. So we don't want um, all, or we don't want contamination from our workflow. No, what we want is to get the target material, the RNA. So para makuha natin siya ng puro, we do extraction protocols. And in this extraction protocol, purity of your target material is being addressed here. So depending on the protocol that you are using, um, kumbaga ito yung mag-dictate kung gaano ka pure yung RNA na makukuha mo. So when we say purity, it means na walang ibang cellular materials na naka-include dun sa ipaprocess mo. Because at the end of the sample preparation, what we want is just the RNA. And then the last concept is the elution and storage. Now, RNA is a very unstable molecule. So, gaya ng sinabi ko kanina, RNA is a biopolymer, no? So, since it's a biopolymer, it's a fairly big molecule, but it's very unstable. So, in the presence of undesirable conditions, RNA will tend to degrade or break down. No? So with that, analysts must be sure that they follow the appropriate protocols. So you use the appropriate liquids to dilute your RNA, and then you use an appropriate container, a clean and a clean container, and then you also do seal the container to prevent spillage, and then place the container on ice. Now, if you don't want, now pag nakakuha na po kayo ng target material or yung RNA, pag nakuha nyo na po yun, and you decide to, say for example, hindi nyo siya ipaprocess agad, you can prevent the degradation of your RNA by storing it in a storage under 20 degrees Celsius. Pag pang matagalan, usually gumagamit na tayo ng negative 70. But usually for COVID testing, most of the samples are immediately processed naman. So one of the extraction kits that we use for analyzing the pathogens in a sample is the MagMax viral pathogen nucleic acid isolation kit. Now, this kit is designed to work with almost all types of samples, including the nasopharyngeal swabs or samples and aspirates. And it's also recommended for all pathogen detection workflow. So not just the SARS-CoV-2. Now, the MagMax uses a magnetic bead technology, ensuring reproducible recovery of high-quality nucleic acids. So, yung contents or yung components po ng MagMax natin ay nandito, shown. So, meron po tayong wash solution, meron tayong binding solution. This is used in conjunction with the nucleic acid binding beads. And then we have the proteinase K solution, and then the elution solution. So I will discuss this in the preceding slides. So how does Magmax extraction kit works? So for the Magmax, um, the collected cells from the VTM and UTM. So this is important. It's not the swab that we are going to process, but it's the VTM or the UTM or whatever media you are going to use. So the liquid part of the sample collection is what we're after kasi nandun po yung cells. So the collected cells are being lysed by the proteinase K at 65 degrees Celsius. So it is at this temperature that the activity of the proteinase K will degrade the cells. So kaya na sinabi ko kanina, we have to disrupt or lyse the cells to get the RNA. Now, at the same time, there are proteins inside the cells called nucleases, and we don't want these proteins or enzymes because they tend to degrade our target material. So, may nucleases na nagde-degrade ng RNA. So, in order to avoid that possibility, the proteinase K will also digest these nucleases. Now, um, part of the components of the Magmax viral pathogen kit is the presence of the chemical guanidine thiocyanate. So this is a component of the binding solution that acts to break down the cells further. And in most studies also, guanidine thiocyanate is um, an agent used to deactivate the viruses. So um, the next thing is that once the contents of the cells are released, the the binding beads incorporated in your process will trap the RNA. So ang the binding beads, pwede mo na siyang ihiwalay from the other cellular materials. So since these beads are magnetized, you can use a magnet so so that you can just separate the beads and the RNA from the cellular materials. Now, the wash solution is also important, and because the washing will help you remove the unwanted cellular materials that were stuck in the binding beads. So repeated washing will further purify the beads. And then finally, the elution solution, which is the part where you are going to uh, dislodge the RNA from the binding beads. So when you use the elution solution, the RNA are then released from the beads. So for the compatible sample types for Magmax kit include the bronchoalveolar lavage, um, the nasopharyngeal swabs, the oropharyngeal swabs, and the nasal swabs, mid-turbinate swabs, and nasopharyngeal aspirates. Now, for the swab samples, we would want to use what is being recommended by our health agencies. So, in order for us to really get the RNA, that the target material from the virus. So this is how your work area in your sample preparation should look like. So basically, what uh, you would want to design your work area wherein mas easily accessible lang yung mga um, consumables na gagamitin nyo. So this one, I will be, I guess I will be explaining this hands-on naman. So to your next week, I will discuss to you each of these consumables as we like try to process certain samples for the Kingfisher and your qPCR instruments. But basically, ito po yung dapat magiging setup po natin. So, kung nakikita nyo, meron po tayong QR code dyan. Pwede nyo po siyang i-scan and you will be redirected to a video that shows how your work area should be established. So gaya po ng sinabi ko kanina, the MagMax kit is a magnetic bead-based technology. So this is compatible with your Kingfisher. My question po dito, are we getting all these consumables from you or we need to order them separately? Yes po, yung consumables, sige, yung consumables po is, meron po kami nito, but sa mga ordering, siguro I will get back to you kung ano po yung arrangement nyo dito with our sales manager. But basically, we also sell the needed consumables for the kits and the instrument na gagamitin nyo po. Okay, so this is how the Kingfisher works. So basically, our instrument, the Kingfisher, it contains a magnet, and it's an automated system. So instead of you moving the magnet into your tubes, you let the instrument do the work. No? So how does it do is that so you have your tube, and contained in the tube are your samples and the reagents of the extraction kits. Now, once the cells are lysed and then the RNA is released, your magnetic rods will go into your reagent or into your tubes, and then get the binding beads which are attracted to the magnets of the Kingfisher. No? And when it does, when pag tumaas na po yung magnetic base, kung tumaas na po yung magnet, basically, matatanggal din yung mga beads doon sa reagent. So sineseparate niya yung beads from the reagents as well as the other cellular materials. Now, the next step is that it will wash the beads using the wash solution and a solution of ethyl alcohol, no? So that it will be your beads will be further purified, and then you would, at the end of the process, you will only get the target RNA. Okay. So yun. So basically, that's the principles of your sample preparation. So we proceed now with the next step, which is the detection step. So gaya ng sinabi ko kanina, as a introductory, um, the, 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 the method of which we detect the SARS-CoV-2 is the use of real-time polymerase chain reaction. So in detail, we use a, a combination of technique, which is the reverse transcription real-time polymerase chain reaction technique. No? So it's a two-step technique which involves one, reverse transcription, and number two, polymerase chain reaction. So sa reverse transcription, ang ginagawa po dito sa technique na to is that once you get your RNA, you would want to convert the RNA into a DNA. So pwede po yun by means of an enzyme called reverse transcriptase. So we know that RNA is very unstable. So in order for us to analyze the viral RNA without any compromises, we have to convert it into a DNA, which is a more stable form of a nucleic acid. Now, once we have converted RNA into DNA, the next step is the polymerase chain reaction. So basically, this technique allows us to to generate multiple copies of uh, the DNA, no, with the use of also an enzyme called the DNA polymerase. So ito na yung issue na, or what if kaya yung na-collect kong samples ay konti lang. No? Actually, we have the saying in molecular biology that less is more. So, ibig sabihin, kahit konti lang po yung na-collect nating cells, we would eventually get a higher amount of our target material with the use of this technique called polymerase chain reaction. So I will proceed with how does the polymerase chain reaction works. So in a polymerase chain reaction, a portion of the DNA, so in this case, from viral RNA, we have the viral DNA na on. So from the DNA, a target sequence is being amplified enzymatically up to 1,000,000 copies. And this reaction is driven by cycles of temperature changes. So, so if you look at the illustration, so say for example, we got initially we have only a single copy of a viral RNA that we have converted also into a viral DNA. Now, what the PCR does is that it targets a certain region of your DNA and then amplifies it or generates a copy. No? And so that at the end of this process, you would end up with two copies. So that's the first, or that's the cycle, the first cycle of the PCR. Now, in order for you to get up to a millions or yes, thousands or millions of copies of this DNA template, also increase the number of cycles for the PCR. So for example, in cycle 1, if you use the PCR technique with only one cycle, guys, let me know if my audio is choppy kasi medyo unstable daw yung internet. So, okay, I'll continue. So, in the first PCR cycle, you would end up with two amplicons. Now, if you use another cycle, in the second cycle, you would end up with four amplicons, four copies of your DNA. At the end of the cycle 3 naman, you would end up with 8 amplicons. So uh, it doubles every time you incorporate another cycle in the PCR step until such time that um, you would end up with a lot of copies of your DNA. So I have here some notes, no, that the PCR is it's a very random process. No? Even though we have a target sequence or a target portion of your DNA, this is very random. So it's not always that uh, the copies are doubled every cycle. No? So um, appropriate temperature and time for each PCR stage is very important. If we have this a different protocol, and then kunyari yung temperatures na ginamit natin for the PCR step is not the recommended, the possibility is that um, hindi po ideal or hindi ma-amplify yung DNA po natin. Ganun. So, aside from that, so kanina for the sample preparation, di ba, one of the important thing is that you have to have a pure RNA. So, this is because in the PCR step, um, there are chemicals called inhibitors which may affect the performance of the PCR. No? And when that happens, you would you won't be able to amplify um, large amounts of your uh, viral DNA. So, okay. So these are the components of your polymerase chain reaction cocktail. No? So we have our DNA template. So ito po yung uh, RNA na kinonvert natin into DNA. No? So, and this will serve as the reference for the DNA polymerase activity. Now, for the PCR, to amplify your DNA template, it needs the building blocks. So, kanina we I discussed to you that the building blocks of the nucleic acids like DNA and RNA is the nucleotides. So for PCR, we use the building blocks, the DNTPs, so that it will be used by the DNA polymerase to synthesize your uh, amplicons or copies of your DNA template. Now, aside from the building blocks or the DNTPs, we have also the forward and reverse primers. So, ano ba tong forward and reverse primers? Ito po yung nagta-target dun sa, kumbaga, we don't want to amplify uh, all of the um, nucleic acids. No, what we want is just a target sequence in your DNA template. So in order for us to have a target, we use a forward and reverse primers. So yeah, so these primers establish the coverage in the DNA template to be copied or amplified. And it also provides the starting site for the DNA polymerase to work. Now, the most important of all is the DNA polymerase, which is the enzyme used to make the copies of the DNA. And then aside from this, we also need the buffer, the magnesium salt, and water, which provide a suitable environment for the PCR to work. So, what about the real-time polymerase chain reaction? So, kanina, yun, na-discuss ko na yung uh, polymerase chain reaction technique. But what about the real-time polymerase chain reaction? So, for the real-time polymerase chain reaction, this is basically just um, doing a PCR technique and at the same time monitoring that process. No? So kumbaga, meron tayong uh, PCR process na nangyayari, then at the same time, meron tayong camera na nagde-detect doon sa process na yon, so that we would be able to know how the PCR is going. No? So for the real-time polymerase chain reaction, um, this uses a fluorescence labeled probes. So ano po ba yung fluorescence labeled probes? So if you look at this sort of basic animation, so you have your DNA template here, and for your uh, real-time PCR, um, we use a series or a series of temperature changes, no, for you to be able to uh, what we call denature your DNA into single strands. So kailangan tong i-denature yung DNA for you to be able to make copies of your DNA. Now, in the real-time polymerase chain reaction, um, you have your primers which bind to a specific region of your DNA, and then you also have your fluorescence labeled probes. So this probe allows you to monitor your PCR reaction. Paano? So as the PCR is undergoing, is ongoing. So you have your DNA polymerase and your primer. So kanina, primer will serve as your starting site for your DNA polymerase to work. No? So as the DNA polymerase is synthesizing a new copy of your DNA, um, it will then encounter your probe. Now, your probe has a structure that is composed of um, an oligonucleotide, which is basically similar with the primers, and your fluorescence dyes. So ano bang function nito? So once the DNA polymerase encounters the probe, no, it will um, degrade the probe. And once it degrades the probe, it releases the part of your probe, which is your reporter dye. No? And once it is released from your probe, it will emit a fluorescence or a sort of a light, no? A signal. So when that happens, then it will able to tell you na the DNA polymerase has worked and there is a new copy of um, DNA that is being synthesized in the process. So now you can correlate the intensity of the fluorescence of those probes with the process of your PCR. No? So in the first cycle, just a small amount of fluorescence is being detected. So that means konti pa lang yung copies na na-generate in the PCR process. But as you go on, from the second, the third, fourth cycle, and so on, you would be able to get a large or a high intensity of fluorescence in your process, and this is being monitored by your instrument. No? So if you would look at this graphical illustration wherein your horizontal line would depict the number of cycles in your PCR, and then the vertical line would depict how much fluorescence is being detected in in the PCR process. So, you know, until such time that the PCR would reach the number of cycles that you have instructed it to do. Okay. Uh, so for the real-time PCR, there is a concept called multiplexing. So kanina ang sinasabi ko, we have to there is the primers will serve as your um, way of targeting a specific region in your DNA. No? Now, um, multiplexing is a process wherein you can target multiple targets in your DNA. No? And this is possible by using um, multiple sets of primers and probes. No? So the way for you to be able to detect, so kunyari may mga gusto mong um, mag-target ng kunyari tatlong regions dun sa DNA mo, then you would use 3 sets of probes and primers. And the way for you to distinguish kung ano-ano yung mga targets na yon is the use of a probe that differs in the reporter dye that is being used. No? So um, depends on your instrument. Say for example for Quant Studio 5, which is your qPCR instrument, it can detect at least um, 6 types of reporter dye. So that means you will be able to multiplex at least 6, not at least, a maximum of 6 targets in your PCR uh, reaction. So, for example, in our detection kit for COVID-19, we have TACPAT um, that uses at least uh, that uses 4 targets: the ORF1AB, the N protein, the S protein, and an internal positive control. And this is being detected all at once in a single reaction. And they are only distinguished by the different dyes that is being used to detect these targets. Now, um, compatibility of the detection kit. Now, for your Quant Studio 5, we have to be aware that um, the detection or the type of kit that you would want to use with your instrument, you have to consider kung yung ginagamit bang dice nung kit na yon ay pasok dun sa range ng detection ng instrument. Sige, bago po ako mag-proceed with because we will be proceeding with how to read the results of your qPCR. So before I proceed, may mga questions po ba? Clarifications? Okay. Okay. Sige. So, sorry. Now, how to read your qPCR result? No? So, after we've done the PCR process, what now? So, basically, di ba, ang real time po natin, what we're after is the monitored PCR run. So, ano po ba yung uh, representation ng PCR run na ibibigay ng instrument natin? This is represented by a graphical illustration called amplification plot. No? So the amplification plot will illustrate the run of your PCR based on the detected fluorescence of the probes. Now, your PCR run in a real-time PCR instrument should look like this. So this is like an ideal amplification plot of a good PCR run. It has a so gaya ng sinabi ko, your uh, the horizontal line would represent the number of cycles that is done in your PCR process, and then the vertical line would represent how much fluorescence was detected by your instrument. Now, a good PCR result or a good qPCR result should have a sigmoid curve in the linear amplification plot. So, um, normally, the first thing that you would want to see in a qPCR instrument is your linear amplification plot. No? So, pag nakita nyo na maganda, na may sigmoid curve, yung amplification plot, then that means the PCR run is okay. No? And that means there is no instances wherein the PCR encountered inhibition or contamination or interference uh, from contaminants or other substances. So you would want to see a sigmoid curve in your linear amplification plot. Now, another way of looking at your amplification plot is the use of a logarithmic plot. So basically, it's just a linear plot wherein the values of your fluorescence is converted using the logarithmic function. So in that way, you would sa logarithmic plot, hindi mo na makikita yung sigmoid curve. Instead, you will be able to see this kind of curve. Yes. So this same pa rin, the horizontal line would detect your, or you would represent your PCR cycle, and the vertical line would represent the fluorescence intensity. But the curve is different now because it's in a logarithmic value. So later, as we proceed with our discussion, malalaman natin kung ano pa yung differences nung dalawang plots. So now in a qPCR result or in an amplification plot, meron po tayong tinatawag na phases in the plots, no? And this depicts the actual PCR run, no? So sige. So in the first phase of your PCR run, meron tayong tinatawag na initial phase. So ano ba yung initial phase? Ito po yung first few cycles ng PCR nyo wherein the detected fluorescence is very minimal. So hindi siya masyadong nade-detect ng instrument natin. And this area of your amplification plot is considered noisy. Kasi and noisy and very unreliable kasi masyadong maliit yung nade-detect na fluorescence. So normally in a good qPCR run, in a good PCR process, you would still have this initial phase. Basically, all of your qPCR runs would have an initial phase of a noisy phase. So then after the initial phase, we have the what we call exponential phase, no? So in the exponential phase, may nade-detect na ang instrument natin na fluorescence. And if we compute it, the fluorescence actually correlates with the ideal PCR process. So ano ba yung ideal PCR process? The ideal PCR process is that at the end of each cycle, the amount of DNA that you have should double, no? And in the exponential phase, na-detect ito ng instrument mo. So if you look at the exponential phase at the linear plot, it's not that well, it depicts as an exponential rate, no? But in the logarithmic plot, it will be depicted as a linear. So later malalaman natin kung ano yung advantage nito, anong benefit nito. Now, after the exponential phase, the PCR would proceed with the lag phase. So the lag phase is a phase wherein may nade-detect na fluorescence ang instrument natin, kaya lang it doesn't represent the ideal PCR run. No? So ibig sabihin, yung increase or yung rate ng pag-increase ng ng number of DNA na synthesize sa ating process is not or is not times 2. So it's not ideal anymore. And this is because your reagents in your PCR are depleting. And when it does, the amplification rate slows down. No, so you can observe it in your plot, wherein in the linear plot, instead na pataas siya ng pataas, nagsistart na siyang nagka-curve, no? And this is also depicted in your logarithmic plot. So parang hindi nagsa-slow down na siya. Ganun. Now, the last phase is the plateau phase. So at this phase, yung nade-detect na fluorescence ng instrument ay umabot na sa maximum. So ibig sabihin, wala nang nangyayaring amplification doon sa PCR run nyo. So wala na or too little. So this is depicted in your plot as a line that is parallel to your PCR cycle or the horizontal line. So nag-horizontal na siya. Ibig sabihin wala nang amplification or konti na lang yung amplification na nangyayari. So at this step, patapos na yung PCR run nyo. Now, of all these four phases in your PCR run, what we are after is the phase that represents the ideal PCR amplification, and it is in the exponential phase. So normally we would want to set a line, what we call as a threshold, in an area of your curve that depicts the exponential phase. So makikita nyo dito sa plot na to, ito yung naka-highlight na area, ito yung part ng exponential phase, no? And this is also depicted here in the logarithmic plot. Now, the difference between uh, the exponential phase area of the linear plot to the logarithmic plot is that in the linear plot, masyadong maliit lang yung area as compared with the logarithmic plot. Now, ano bang advantage nito? Kasi we would want to set a threshold in our qPCR run. And the threshold would um, indicate at which part of your qPCR run would mean that the detected fluorescence is just a noise or a background as compared with the detected fluorescence that is really a true amplification. So we have to set a threshold on that plot, and the threshold is always horizontal to the PCR cycle. Now, yan na yung sinasabi ko. Mas magandang mag-set ng threshold sa logarithmic plot kasi mas malaki yung area na pwede mong pag-decide ng threshold kesa sa linear plot kasi masyadong maliit. So, how would you know, or where would you want to put your threshold in your plot? So in the logarithmic plot, the part wherein there is a linear, a straight line in the curve, that is the area wherein you can set your threshold. So aside from the threshold, which distinguishes the background fluorescence from the true amplification fluorescence, we have also the concept of what we call a CT value. So yung CT value po, ito po yung CT value, or it means threshold cycle value. So this is the PCR cycle at which the amplification curve will intersect with the threshold. So after we set the threshold line in your amplification plot, the part wherein the amplification curve intersects with your threshold is your CT value. No? So this just means that at this point, yung na-detect na fluorescence ng instrument nyo is no longer a background or noise. Ibig sabihin, true detection na siya. True fluorescence detection na siya. So, ano bang importance nito? Yon, CT value will tell you how much sample you have before PCR. So, ano bang ibig sabihin nito? So, if you look at your amplification plot, you have set your threshold, and you find that your curve seems to be at the earlier cycles of your PCR run, then that would mean the concentration of your DNA in your sample is very high, so that maagang na-detect ng instrument yung fluorescence. Okay? But if pag konti lang yung DNA na nandun sa samples nyo, then your curve will appear at a later stage or at a later cycles of your PCR. No? So, paano mo siya malalaman, this is because of your CT values. So if you have a lower CT value, then that would indicate a strong positive detection of your target. So inverse yung relationship niya. Pag mababa yung CT value, ibig sabihin mas maaga yung fluorescence na nade-detect ng instrument, which means mas madami yung DNA na nandun sa sample niya. No? Pag mataas naman yung CT value, ibig sabihin medyo late yung pag-appear ng fluorescence. So ibig sabihin weak yung detection na nakuha ng instrument natin. And this process or this concept of the 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 strength of your detection is what we is what does what the limit of detection states, no? So for the limit of detection, this is actually the minimum viral concentration that spikes positive in a qPCR result. So ano ba ibig sabihin nito? So pag ano yung pinakamaliit na concentration or na amount ng DNA na nade-detect ng instrument nyo. So, okay. So for example, in our kit, the TACPAT COVID-19 combo kit, the limit of detection, which is the smallest amount that can be detected by the qPCR instrument, is at 10 genome copy equivalents per reaction. So, ibig sabihin, 10 copies, at least 10 copies. Kung meron kang 10 copies doon sa samples mo, madedetect siya ng instrument. And this is represented by the CT values here. So, for example, in your Quant Studio 5, no, at 10 GCE per reaction, you would be able to get the CT values at 29, 30 to 31. So ganon siya ka sensitive. So apart from the amplification plot, apart from the CT values and the threshold line, we also have the concept of baselining. So ano ba itong baselining? So di ba sa lahat ng qPCR results natin, meron talagang part ng PCR na noisy. And that is what we would want to remove from our result. And the process of removing that or letting the instrument know that this is just a background noise is baselining. So usually, the PCR cycles at 3 to cycle 15, ito po yung mga tinatawag natin na noisy cycles. So, dito natin sineset yung baseline natin. Now, this is important because correct baselining will improve the curve that is represented in your amplification plot. And so we would want to make sure that the baselining that we set in our amplification plot would be just at the part wherein or just the part before the amplification has been detected. So, for example, in this amplification plot, we would want to set the baseline at the cycle 3 to cycle 15. Kasi after that, may steady detection na nung amplification or yung fluorescence. Now, another plot that you will encounter in your qPCR instrument is your multi-component plot. So, the multi-component plot is the sort of the raw plot of your qPCR result, no? And it shows the complete uh, spectral contribution of each fluorescent dye during the PCR run. So, usually, ginagamit natin ang multi-component plot to verify whether the detected fluorescence is really uh, a true detection or a false detection or no detection at all. No? So, kunyari, for the negative sample, makikita nyo na true negative siya pag ang multi-component plot niya is just a steady line, a straight line, a horizontal line for all of your targets. So ito dapat yung makikita natin for a true negative sample. And then for a positive sample, you would want to see a sigmoid curve also on the multi-component plot. So that means there is really a true detection of fluorescence in your samples and not just a glitch in your instrument. So malalaman mo yan pag dito sa multi-component plot may nakita kang ibang curve aside from the sigmoid curve. So that means there is something wrong with your qPCR run. Now, the common errors that we can encounter by checking the multi-component plots are stated here. No? So, usually, pangit yung curves na makikita natin sa multi-component plot pag nagkaroon po tayo ng pipetting error. So, ibig sabihin, yung mga volume ng reagents na necessary for a PCR run ay konti or kulang, no? And this will affect your plot as well. No? So aside from pipetting errors, so this is also important that your pipetors, the liquid dispensing instruments that you have should have should be calibrated as well. Aside from the pipetting errors, we also can see in the MC plot that there has been an evaporation that took place in the PCR process. No? And this is important kasi in your, say for example, in your plate wherein you put your samples and then you put it in the instrument, no? Ang qPCR kasi is a PCR technique as well. And it uses a set of temperature changes na umaabot usually ng 95 or 94 degrees Celsius. So it's a bit high. And this will cause your liquid to evaporate. No? Now, once the liquid evaporates, no, it will somehow distort the way that the instrument will detect your sample. So para siyang nagfa-fog doon sa camera ng instrument nyo. So we would want to avoid that. So paano? We can avoid it by using an appropriate containers, or the sample plates that we would want to use should be appropriate. And then we would also want that the PCR conditions that we are using in a qPCR instrument should always be appropriate also. No? Too much of the temperature will cause your plot to be pangit. Ganon. So aside from evaporation, we have the bubbles or droplets. So ito din yung isa sa problema. So sa MC plot, nakakakita tayo ng pangit na curves because there's some of the samples that has bubbles in the in the liquids, no? And this will also distort the way that the instrument will monitor the PCR run. So we would want to avoid that. So ang ginagawa natin is usually we check bago natin i-run yung mga plates natin or yung mga tubes kung saan nandun yung reagents at saka yung sample natin. So we would want to check those containers. Dapat walang bubbles doon sa liquid. No? And that yung liquid, hindi siya sabog. Ibig sabihin, hindi siya doon yung may mga liquids na dumidikit doon sa ibang parte pa ng container. So we would want to make sure that all of the liquid is at the bottom part of your container. So aside from the multi-component plot, we have the raw data plot. So yung raw data plot, this is just to show you the total raw fluorescence in each filter pair at a given cycle. So for most of the COVID detection kits, multiplex na kasi ang ginagamit. So they involve more than one target, no, to detect the SARS-CoV-2. So malalaman natin na tama yung mga nade-detect ng instrument by looking at the raw data plot, no. So medyo kakaiba yung raw data plot kasi yung horizontal line niya, this would mean the filter channels in your instrument, no. So for your Quant Studio 5, meron tayong a maximum of 6 channels, right? So in the raw data plot, meron din tayong 6 channels dyan. Actually 12 because each channel is we have X1 and M1. So for example, you have your dye at the kunyari, yung detection kit na ginamit nyo has a dye that is detected at the X1, M1 channel. So kung may positive yung samples nyo, eventually yung raw data plot nyo, dapat meron kang makitang increase ng amplification doon sa X1, M1 channel. So for example, in this raw data plot, this is the raw data plot at the first PCR cycle. So makikita natin na medyo mababa pa yung nade-detect na fluorescence ng instrument. But at the last PCR cycle, medyo tumaas na yung na-detect na fluorescence for each of the filter channels. So ibig sabihin, yung dyes na ginagamit natin ay belong doon sa X1 M1 channel. We also have the dye at the X2 M2 channel, X3 M3 and X4 M4. Now, ano bang importance nito? Pag yung dye channel kunyari ay mali, no? Mali yung pagkalagay natin na dye doon sa instrument. So, kunyari ang ginamit natin ay FAM dye, pero ang nilagay natin sa instrument, ang sinet natin sa instrument ay ibang dye, then the raw data plot would tell you na ay mali pala yung dye na naset mo. So, for you to correct it, actually, you can just reanalyze your qPCR result without rerunning the PCR. No? So you just have to look at the raw data plot and ensure na tama yung mga dyes na na-set natin dun sa qPCR instrument. Okay. Now, for your Quant Studio 5, ito yung sinasabi ko na filter channels. No? So the filter channels is usually depicted in colors. So we have the blue, green, yellow, orange, red, and the deep red. Sorry. Now, one thing of one thing good about your Quant Studio 5 is that it is a pre-calibrated instrument, meaning that some dies have already been calibrated during the manufacturing process of the instrument. So the pre-calibrated dies are listed here. Now, if you would if you are going to use a detection kit that um, that uses a die that is not listed here, no, we have to um, calibrate it, no. But normally for Quant Studio 5, kung ano lang po yung uh, custom dice, yun lang po yung pwede nating i-calibrate. So for example, if your detection kit uses a Texas red die, we have to calibrate the Quant Studio 5 with a Texas red die. Also, um, this is important because yun nga, yung compatibility ng detection kit natin magdedepende dun sa filter channel ng qPCR instrument natin. Now, if you use a die, say for example, if you use a die that has an excitation wavelength that is more than 700, no? Ibig sabihin, out na yon or wala na siya dun sa range uh, ng filter channel ng qPCR instrument natin. Okay. So yun. Just to summarize this, no. I hope if you have questions also, you can just chat me or um, you can turn on your mic and ask questions. But in summary, no, for you to have a good qPCR result, no, you have to have a positive and negative controls. This is really important kasi this would tell you kung maganda ba yung run ng qPCR nyo o hindi. If your positive control, if the result of your positive control is not good, then this would serve you a reference na there is something wrong in your qPCR run. If you have a detection happening in your negative control, ibig sabihin may nade-detect siyang viral viral DNA doon sa samples nyo. Ibig sabihin there is possibility of contamination in your samples. So importante tong controls. It should always be incorporated in each of your qPCR run. Then you also need to ensure the correct labeling of your fluorescent dyes. So ito na yung sinasabi ko kanina. So when you mislabel your fluorescent dyes in your instrument, it would always result to a very ugly, very ugly qPCR plot, amplification plot. And pero don't fret because we can reanalyze the qPCR result naman. We have the design and analysis software which I will be training you by next week. And then another one is to always check on the multi-component plot for possible variations during the PCR run. So, yun, pag may mga bubbles or evaporation that happened in your PCR run, you will be able to tell them at the multi-component plot. And in that way, you would be able to know how to interpret well your qPCR result. Next is to check the baseline and threshold of the amplification plot. If you are not satisfied with your baselining, kasi for your for our software, the design and analysis software, which comes with your Quant Studio 5, meron po tayong tinatawag na automated baselining and threshold setting. But if pag tiningnan nyo yung qPCR plot nyo or yung amplification plot nyo and you think na hindi siya maganda or hindi appropriate yung baselining values and the threshold na sinet nung software, you can adjust them manually. Provided that you also have a basis of your why did you adjust them. Now for difficult qPCR results, and which is really unavoidable kasi I mean we are dealing with very varied sample types, and then ibang tao pa yung tinetest natin. So most likely meron din tayong mga very difficult qPCR results. So when that happens, you can always contact us, the field application scientists, so that we can help you how to proceed with your data analysis for that one. So for troubleshooting, kung may problema yung mga instruments, you can contact us. If you need further help on how to interpret the qPCR results, the plots, no? not the report, the yeah. So, but just the troubleshooting, you can contact us, no, further, and we will be more than willing to help you. Okay. So, yun. So that ends my lecture. So, are there questions? Yes po. Hello? Sige po. Yes po. So, um, normally, although for the software of the Quant Studio 5, medyo maganda na kasi yung automation ng data analysis ng software. So, bihira lang din yung kailangan yung mag-intervene dun sa software. But if you really have to, provided that you see that the amplification plot is really very ugly, you can adjust manually. So it's in the software. It's kind of hard for me to train, but late next week I will show you how to do the manual adjusting of the baseline and the threshold. I would say 10. Yeah. Yeah. No. Uh, uh, no worries, sir. This is also, I think you can also gain based on experience as well. So pag meron na kayong mga samples talaga sa lab nyo, then you repeatedly analyze the plots, then you will be able to really understand it. But for now, um, yeah. you can rely with the automated baselining and threshold of the software. Kasi maganda talaga siya. Okay. So, any questions pa po? So I will also share with you my presentation. Um, yeah, I will share with you my presentation today so that um, at least bago tayong magkita-kita by next week, you can review the the qPCR plots that is important for data analysis. Um, any more questions? Ayan, may nag-chat. Sige. Okay, no problem. Sige. Any other questions po? Um, sige. Before we end this discussion, I also would like to um, just to check kasi I mean this is also important bago tayo mag-proceed or bago tayo mag-conduct nung hands-on training next week. Uh, I would just want to check with you this one. Nakikita niyo po ba yung uh, so nakikita niyo po ba yung screen ko? Okay. Uh, setup natin. No? Ito yung mga instruments and mga uh, other equipment na kakailanganin natin for the qPCR detection workflow. So I can share you this one as well. I will send it to you on your email and then you can get back to me kung ano yung mga uh, wala sa lab nyo. And I will um, since we are also dealing with Kingfisher, right? And Kingfisher is actually an instrument that offers you a high throughput. So ibig sabihin, madaming samples yung mapaprocess nyo in just one run. Now, for you to be able to, because the instrument is not fully automated, there are steps in your Kingfisher that needs manual dispensing of the reagents into the plates. No? So para mas mabilis yung pag-dispense natin ng reagents, kailangan natin ng multi-channel pipetors. No? If kung wala naman tayo, we can do with the single channel na. But if you have multi-channel pipetors, that would be better. Mas mapapabilis yung pag-dispense ng reagents natin doon sa containers. And yun. So I will share this to you on email and I hope you will get back to me kung ano yung mga kulang natin and then we can find a way to make ends meet. Okay. So ayun. Do we have any more questions? Oh, I see. So okay lang din naman na may na single channel. Yun. Pwede naman yung single channel. Okay. So, ayun. So, I hope na uh, madami kayong natutunan sa discussion natin. And I am always available if you have any questions na regarded dun sa na-discuss natin. And yun. So if there are no more questions, yun, I will end this discussion. And thank you po for listening and attending this meeting. And ingat po kayo and ayun. So thank you. Salamat po. And I will thank you. I will end this meeting now.